much of the project was geared toward flying up and down individual members of the platform, identifying the range of Lophelia, looking for large colonies for detailed digital still photos, and then sampling.
This study was one of the first of its kind. We're here looking at cold water corals that attach to oil and gas platforms in the Gulf of Mexico, and our primary focus was Lophelia pertusa. It's a white coral, it's a scleractinian coral. It forms on hard substrates in the deep Gulf, and we were looking to see to what extent the oil and gas platforms provide habitat for Lophelia, and to see if we can glean any information about their depth constraints, because oil platforms provide a nice sort of structure for us to look at what depths they occupy, and if we can understand what their tolerance is related to depth and therefore temperature and other environmental factors that we're measuring.
The ROV is a system we built in-house. We call it Kraken 2 or K2. It's a 1,000-meter science class ROV, 1,000-meter depth capable. We have a number of payload capabilities on board for science, including quiver samplers, tube core samplers, bio boxes, suction samplers, and other things. And it's an imaging device. So there's high-definition video cameras, paired lasers, digital still cameras, and an assortment of other devices for collecting oceanographic and optical data.
There were a couple different groups. The first group that we had was from Penn State University. They were responsible for doing all of our imaging. So that looked at taking HD video and scanning the vertical members of the platforms, looking for density, distribution, and also taking digital stills to calculate growth rates of the corals. So the idea is that if we know the age of the platform and we can calculate the distance that the coral grows outward from the platform, we can calculate a minimum growth rate. This has been done on a number of platforms, mostly using industry video, whose intent is obviously not to count corals or estimate their growth rates, but this was the purpose of our cruise, to look at that. We also had some scientists from the U.S. Geological Survey, a couple different projects going on there. One was to collect samples of Lophelia to look at the genetic connections between different platforms that are obviously spaced apart over the Gulf, and we can get some information about gene flow. Also, we had a scientist that is looking at the microbial communities that live within the Lophelia polyps. And we also had a group from USGS that was sampling the sediment to look at the myofauna communities.
So we're taking box core samples to get background sediment for our macrofauna analyses. It's basically a complement to the samples we'll be taking using the ROV. The box core is deployed and it takes a sample off the bottom, and then we go in and use tube cores to subsample the box core to get an idea of the organisms that are living in the sediment off of our platform. For each box core we take two tube cores. One is used for macrofauna analysis, and so back in the lab they will sieve that material looking for all of the organisms and fauna living in it, such as polychaetes, bivalves, gastropods, and then the other core we use is to do sediment chemistry and particle size. And so we'll look at the redox oxidation, and we'll measure the salinity of the pore water. And then back in the lab we also have a process that will look at the percent of different grain sizes in the sediment, and that all can be factored in to when we look at the macrofauna analysis to see what kind of critters are living in that habitat.
So once we get the tube cores from the box core, we're going to put the tube cores through an extruder, and we're slicing at 3 different intervals, at 0 to 2 centimeters, 2 to 5 centimeters, and 5 to 10. And basically each of those fractions is going to be its own sample, and then back in the lab we're going to be looking at them under a microscope to count and identify all the animals that live in those fractions. We use the sieves back in the lab to separate the two size classes. Macrofauna is greater than 300 micron, and the myofauna is greater than 45 micron, and we sort both of those independently. To compare to the background samples, we take push cores down at the bottom for near the coral or near the platforms. And that's for the same kind of analysis that we do with the box core samples to compare the community composition and abundance and biodiversity at both areas.
What happens when we take push cores with the ROVs? The ROV goes down and it has a push core with a little T handle, and the arm will extend and slowly push the push core into the sediment, and then it'll do 6 in a row. That way we prevent any possible contamination, and when you pull it out and some sediment may fall. And then we'll collect those 6 push cores and put them back in the quivers and bring them back to the surface for processing.
Our group from Temple University, we're interested in water chemistry parameters related to ocean acidification. And we have another graduate student that is interested in calculating growth rates using radiocarbon dating.
Primary challenges on this were involved working around the oil platforms. First off, we had a lot of challenges due to the fact that we had minimal information about the structure of the platforms and any known hazards or obstacles around the platforms. So that was first and foremost a difficulty. The second one is just working around anything that poses hang hazards is always technically difficult. And the third primary thing was working mid-water. Anytime you're working mid-water, you don't have a bottom fix when you're piloting the camera. You're looking ahead, but if you get pulled off a little bit, you don't have a frame of reference other than your instruments that you have within the vehicle. So it's sort of flying by instruments as you would in a plane in a fog condition.
Yeah, the coolest thing for me was the dive on the Ram Powell platform, which is 1,000 meters deep. And we found Lophelia at 800 meters there, which to me was really interesting because I've been studying Lophelia for 4 years now, and this is my 6th time to the Gulf of Mexico, and we've only ever seen it between 300 to 600 meters. So we really sort of just considered that the depth limit or depth range was 300 to 600 meters. So breaking that construct was really cool, and it was really exciting for me to see that.
K2's previous maximum depth was 898 meters. When we visited the Ram Powell platform, we hit 1,014 meters to the bottom. So that was it was nice to see the vehicle actually get to the depth it's designed to work at. So that was kind of a highlight, that's for sure.